Okay, so I think it's time. Hi, Lily.
Hi, nice to meet you.
Yeah, nice to meet you. So welcome to our last session, data mining and statistical methods. So my name is Hsiang-Yun from Tübingen, Austria. So in this session, we will have two invited talks, three paper talks, and one abstract talk. So let us enjoy this fantastic session.
Great, thank
And
you.
And first of all, I would like to introduce our first speaker, Dr. Lili. She is the Vice President of Clinical Informatics at SEMA 4 and an Assistant Professor at the Econ School of Medicine and Mount Sinai in the Department of Genetics and Genomic Science. She has a lot of contributions after I investigate her website, and not only in academia, but also in industry. Due to our tight schedule, I will leave the time to Lily and to show us her recent investigation about COVID-19. Let us welcome Lily.
Great, thank you. Can you see my slides?
Yes.
Okay, great. Thanks very much for the introduction, and thank you for this opportunity for me to talk about our recent findings in COVID. We have found the racial disparities and identified the prognostic factors in the COVID hospitalized patient population based on the EMR data from Mount Sinai Health System in New York City. Before I start, I would like to declare that this study is exempt from IRB. This is based on de-identified EMR and is not considered as a human subject research. Patients or the public were not involved in the design, conduct, or reporting or dissemination plans for our research. As we know that over the past 6 months, we have been experiencing the world struggle with the COVID pandemic. This animation shows the number of the confirmed cases in US by state in early months. Healthcare professionals are being stretched to their limits to identify optimal treatment to lower the mortality rate and improve recovery. New York State, especially for New York City, remained the US epicenter for of the pandemic at the early months to April, with roughly 20% of the cases and fatalities in US. The first positive case was reported in our hospital on February 24th, and around the first week in April, we reached the peak for mortality cases. After that, the numbers were decreasing, and we have flattened the curve. The second sharp peak here indicates the death after massive COVID testing. So our study reports the data from the first case to mid of April. I would like to give you an overview of our Mount Sinai Health System. This is the largest academic medical system in New York City, encompassing 8 hospitals, a leading medical school, and a vast network of ambulatory practices throughout the greater New York region, with more than 3.5 million outpatient visits per year. In this study, we focused on our 5 member hospitals, which use EPIC system, including hospital in Queens, Brooklyn, and 3 hospitals from Manhattan. Our EMR is the largest comprehensive EMR, representing the population and the racial diversity in New York City. We have 8 million unique patients in EMR and 5.2 million with comprehensive clinical data, including vitals, diagnosis, labs, medications, procedures, and clinical notes from 2003 with over 15 years follow-up. So given the high mortality rate in New York City and the uncertainty regarding COVID progression, accurately evaluating prognosis for mortality and discharge among the hospitalized individuals is critically needed. Our patient cohort is from February 24th to April 15th, about 10 days after peak of diagnosed cases. At the moment we started the study, it was the largest and the most racially diverse US-based COVID case series. We have more than 6,000 PCR confirmed positive cases by Roche Compass 6800 to detect for SARS-CoV-2 virus, and among those, inpatients are 3,273. We then collected clinical features, including patient demographics, vital signs, and lab measurements at admission as a baseline, chronic conditions, medical history, and social behaviors to assess three outcomes: deceased or discharged to home or other locations, but not concerning intensive medical care, and continued hospitalization as rate censored. We use statistical methods, including multivariate logistic regression, competing risks, survival analysis to analyze the mortality rate and the discharge rate over time. Competing risk models are recommended over Kaplan-Meier survival for with multiple underlying causes. The goal is to estimate the CIF, is called the cumulative incidence function, of inpatient, in-hospital death and discharge for each individual cause K for a patient who stayed at hospital for a duration of a TF. We also performed the multivariate analysis to estimate the contribution of each potential covariate to the cause-specific hazard using the Cox proportional hazard approach by treating one cause as an event and the other as a rate censored. Those functions are all available in our package. Overall, we have a 23% mortality rate in our cohort. We found African Americans and Hispanics were overrepresented in the COVID positive cohort, accounting for about 25% and 29% respectively, among all the infected patients with known self-reported risks, which were both significantly higher rates than expected based on the population-based rate, which is 19% and 12% respectively, even after adjusting for age. Well, tested cases are not lower in African American and Hispanics. And in-hospital mortality did not further this risk disparity, which still remain 24% and even lower for Hispanics as 25%. We also noted that age distribution of COVID patients within each race were different relative to our source population. For example, a white deceased cohort shows a different age distribution from other race groups. The density increased continuously as age increased, forming a triangle shape. While African Americans show the highest density at the age of 74 years. Older age increased the risk for mortality, which is well known now. For middle age group, 10% to 20% mortality rate. But for older age of 80 to 90 years, mortality rate reaches 40%. Age above 90 were de-identified to 89 due to PHI. So importantly, we found non-significant differences in age-adjusted in-hospital mortality rates across all racial groups. This was estimated using Bootstrap by sampling the patient in that rolling 10-year age window 500 times with a 95% confidence interval reported. We focused on COVID inpatient with at least 1 day in the hospital. So we summarized their demographics features, vital signs, and the lab tests at admission, which were used for assessing mortality and discharge. This is also a good way to QC the data and identify the patterns. We estimated the CIF, the cumulative incidence functions for in-hospital death and discharge using competing risks survival analysis. In our full cohort, we estimated that by 10 days post-diagnosis of COVID, patients have a 51% chance of being discharged from hospital and a 22% of chance of in-hospital death. By grouping our cohort using different demographics and hospitalization factors, we found the differences in time-adjusted mortality among different racial groups is more enriched in whites due to its older age group, but no differences in the CIF of the discharged patient. We also found that females have better outcomes compared to males. They trend towards lower mortality rates and significantly higher discharge rates. Hospitalized patient outcomes are also differed by hospital and care area types. We found that patients admitted to a hospital in Brooklyn and Queens experienced significantly worse outcome, the orange and blue lines above. Mortality is higher, discharge is longer compared to other 3 hospitals in Manhattan. This could be due to relative older COVID patient in Brooklyn and Queens. As expected, we also observed dramatically worse outcome for all patients admitted to ICU compared to other care units. Identifying which patients are at highest risk for the inpatient mortality during admission will help and also enable clinicians to target interventions, allocate resources, and then make more informed triage decision. So we first aim to identify etiologic factors associated with death and versus discharge among inpatient. Risk factors for death included advanced age, maximum temperature during hospitalization, higher respiratory rate per minute, ICU care area, higher white blood cell counts, elevated serum creatinine, which is a marker for assessing kidney function, and higher ALT for assessing liver function. We also found patients with higher temperature, higher oxygen saturation at admission, as well as those with a higher minimal oxygen saturation levels during hospitalization were more likely to be discharged. Then we employed a repeating risks survival analysis to estimate the hazard ratio of covariance on both mortality on the left and the discharge on the right. The cause-specific hazard models showed advanced age significantly increased the risk of mortality and decreased the probability of discharge. Similarly, care in ICU, white blood cell counts, elevated creatinine, and ALT all increased the risk of mortality and prolonged the hospital stay. BMI increased risk for mortality, but not prolonging the in-hospital stay. While patients with asthma had a longer in-hospital stay, but not significant risk for mortality. The higher oxygen saturation in hospital shortened the inpatient stay. Furthermore, we did not find an association between risk and the clinical outcome after adjusting for underlying covariance, which further suggests racial parity in terms of in-hospital mortality in our cohort. In summary, we comprehensively summarized the clinical characteristic for COVID patient through the peak of the pandemic in the greater New York City area using one of the largest EMR system. We demonstrated a higher age-adjusted rate for COVID infection in African Americans and Hispanic compared to their overall population. However, outcomes for the hospitalized patient showed the racial parity after adjusting for other clinical factors. And we also identified older age, lower oxygen saturation, elevated respiratory rate, and elevated lab parameters as clinical relevant prognostic indicator for increased risk for mortality for hospitalized patient. We also developed a prognostic model to predict the outcome for inpatient based on the baseline data. Due to time limits, I did not show it here. Feel free to check out our paper deposited on medRxiv. The updated version is under review. This is my clinical informatics team at Semaphore. Semaphore located in a beautiful harbor at Stamford, Connecticut. My team mainly focus on data-driven digital phenotyping development and building tools, algorithm for deep learning on imaging for prognostic and diagnosis, machine learning modeling for disease prediction, and enable us to launch a digital clinical trial to validate our algorithm and the tools. So we aim to deliver and deploy the clinical applications. We are expanding, and I have several openings on data scientist position. If you are interested, please go to our website and apply the job or send an email to us. And thank you for your attention.
Okay, thank you very much, Lily, for the very interesting talk. Do we have any questions from the audience?
Yeah. Hi, Lily.
Hi.
Hi. Say hi to Ron.
Thank you.
Yeah. Yeah, I read your paper when you deposited in the medRxiv with a lot of interest. So it seems that you're suggesting the disparity in COVID is mostly on the social demographic nature of African Americans and Hispanic, or where they get treated. Once they receive the standard treatment of care, the disparity disappear.
Mm-hmm. Yeah.
I wonder if this is related to earlier data, because we have data here at the University of Alabama at Birmingham Hospital. We also observed that disparity, and we saw that the disparity not only exists in those standard of care, access to care, but also the strong correlation between obesity and Hispanic population. And that obesity is really worsening the disease progressions. I wonder if you didn't look at that, or maybe this is earlier data, you didn't find a similar trend?
For the obesity, we have a BMI, as I showed earlier, right? Actually, I think this is for the early, right after peak, right? Because we have not considered whether the patient could be readmitted to hospital. But given the fact that in Brooklyn and Queens, where the most population will be African American and Hispanics, and towards a low social economic status, I think they do not have a ability to enable them to work from home, so they be exposed environment that with a very load. Due to some maybe they prefer living close to each other, or they do not have a self like a PPE, or can have a social distancing issue. So I think that which reflect our the early findings to explain why the infection rate is are higher, right, for these African American and Hispanics. But you're absolutely right, which is actually we want to bring that message, standard of care, or the care offered from our hospital did not further this disparity. We treat every single patient are same, and because the age tend to be a little bit younger for these two race, I think I certainly diabetes and obesity, higher BMI, even not necessary to be I mean the high blood pressure at the time of the admission, a higher risk for those population. We have not take a further look, although this is an early paper. The data is only collected at the baseline, we of course, have so many factors need to be collected, including all the medical history, right, as well as inpatient treatment to better assess and build a profile, build a predictive model for outcome.
Yeah, another question is, your company is doing a lot of sequencing work. Are you adding sequencing, for example, the MHC regions of the patients or the virus strain to actually do some predictive work?
Yeah, that's actually underway. We actually indeed, not only just for sequencing, but we also collect, Semaphore, we have our own testing kits. And my team right now is also working on, contact tracing for collecting some device data, Bluetooth data, as well as if, we have some sequencing data altogether. To try to build a comprehensive model, either to predict or to assess the probability can come back to work, or the risk to get the COVID positive.
Thank you.
Thank you very much. Actually, I have a quick question since I'm not coming from the medical domain. I'm from the visualization community. Could you give, for example, it's usually the case that's very difficult for us to get interesting data because we are not working very strongly, not in the hospital, for example. So could you give us some guidance how we can get such kind of sensitive data? Besides, do we only have the choice to collaborate with people from the hospital or the company, or is there any chance that you open this as an open source data resources?
As far as I know, the EMR data will not be shared, even between institutions, even if it's de-identified. But I think you can maybe set up some collaboration, right? If you have some hypothesis or question want to solve, and you can work with with us or with some with the academic center or hospital can, I think, set up the collaboration will help you. Another thing is you can get some public domain data set, like from surveillance data. So it really depends on question, if so what question, right, you want to try to answer to get what type of the what level of the data.
In our field, usually we also develop a kind of explorative approach. That means we don't know what's inside the data, and we want to develop an approach that support user to explore the data to somehow find out something important within the data. So maybe at the beginnings, even the analyst doesn't have any hypothesis or assumption. So we need some real world data set for us to do a kind of trial and error. That's why I'm asking. This would be a very interesting data set for us.
Yeah, I see. Yeah, actually for us, I think we're trying to solve the questions that the clinicians bring up, right? And they want to improve the outcomes. So this is maybe a little bit different. So we have a question, then we locate the data, and then we do the analysis, or we design the study. Yeah.
Thank you very much. So I think, I think we have to. Any questions? A short one, because now we are running out of time. Okay, if there is no further questions, let us thank Lily, and we will move to the next presenter. Okay, thank you, Lily.
Thank you.
Okay, so our second speaker will be Dr. York mentioned, and he's currently a full professor at the University of Vienna and a researcher at the Research Center for Molecular Medicine of Austrian Academy of Science. So he specialized network theory in bioinformatics, and today he will present us a very cool way, I would say, to handle and interact with large molecular networks. So let's welcome Josh.
Hi, thank you very much for inviting me. I was really looking forward to actually going to San Diego. I've never been there, and it is such a pity. But it is still cool that we can do this remotely. Thanks again for having me. So I will be talking about something that is a bit different, I guess, and what I really want to show you is this. So what I would like to show you is to introduce to you, let's say, our vision of how we think we can explore and work with really large and complicated data in, I don't know, let's say in the intermediate future, if not now, but in order to introduce to you first why we think that is a good idea to begin with, I will actually be talking a little bit about networks in biology and medicine, which is really our core business, and then talk second about our virtual reality platform. And actually I will be trying to do a live demo, which is again first virtual conference, first time I'll be trying to do this live, so please bear with me. This is not as smooth as I hope it would go. So to us, networks are really our core business, and we are intrigued by them, or we think that they are important and make sense because they are a beautiful representation for any kind of complex system that consists of many interacting components. And of course, in biology, such systems are everywhere, from the biomolecules that would be interacting in our cells that are built the molecular machines that really are the mechanistic basis of any kind of biological functions, to cells that talk to each other, let's say in a brain that send biochemical signals. Also organs can exchange all kinds of signals, all the way to humans that can exchange viruses. So we have a global epidemic, and all of these things are intrinsically described by networks. And of course, networks are useful. So they are not only prevalent as not to describe a system, but to actually derive knowledge from it and how it really works. And I will give a few examples just to indicate that there is a very intimate relationship between the structure, so the connectivity patterns that we can quantify and observe in networks, and what they mean in biological terms. And the first example is a classic paper that's almost 20 years old, that was the first one to show that the importance of individual genes or gene products for an organism is intimately linked to how central the particular gene is in a molecular network. So it's a bit hard to see, but the red nodes here represent genes that, if knocked out, are actually lethal to yeast colonies, and they are all, or they have a certain tendency to be located in the middle, whereas those genes that are more redundant have a tendency to be actually at the periphery of these networks. So there is a very strong relationship between the location and the biological importance. A second example is that also network structure that look like very densely connected local clusters have a particular biological meaning. And in this case, this is a beautiful paper from Constanze et al., where the network is actually a genetic interaction network, and you see that these very dense little clusters, and they actually correspond to biological functions. So all these genes doing something together in a network sense, they would be these dense little communities. This is not only true for functions, so also dysfunctions or diseases have their own network or connectivity patterns. They are a bit broader, but they are also not scattered randomly in these molecular networks, but they aggregate in what we would call disease modules or disease neighborhoods, and a beautiful correspondence between something that is very molecular, that is how overlapping two modules would be, to something that is really a population scale quantity, that is how often would two diseases co-occur, is actually in this network-based overlap. So there is yet another connection between very, very microscopic molecular entities all the way to population scale biomedical quantities. And the last example that I'd like to bring is from a group here from my institute from a couple of years ago, where they found also very intriguing global structures in biological networks, where this lipid co-regulation network that has this very peculiar round shape it's not entirely clear where it comes from, but our best guess at that point is that it is a natural consequence of the fact that a cell returns to homeostasis after being perturbed in some sense, and that these the different steps of co-regulations are actually forming this ring in this very abstract space. So, as you, my peers, are certainly well aware, these pictures are all very convincing, but of course the reality of how we really work looks much more like this. So it's a lot of data analysis, we do a lot of coding, it's a lot of statistics, it's a lot of math. So the question is, how can we actually also for large-scale molecular network structures make use of this unique feature that networks have, that they have an inherent really visual quality to it. So while all these four examples were a little bit, post hoc, where the visualization was actually, I guess at least in 3 of these 4 cases, done afterwards to illustrate a point that was in fact found by statistics, I think there is a lot of potential to actually try to do it the other way around. So what if we could use these networks, and what if we could use visualizations of very large scale data structure to really form hypotheses, to speculate on what are meaningful patterns, and really do it the other way around. And we got inspired in our question of, how would we imagine that the data analysis of the future would look like? What would artists do? And I feel that they have already many years ago came up with what I think are quite intriguing solutions, and that is immersion. So the interface to a very complicated data set, or also the interface to a very powerful data analysis engine, is not a small window into looking into a computer screen, but it is really an immersed 3-dimensional environment where I would be manipulating objects or even code as I would in my very natural 3-dimensional environment. And I also think that the artists, regisseurs, the directors of these movies, they got a second thing right, and that is that ultimately the hero in all these stories is the human. So at least from my own experience, there are certain pitfalls in collaborations between medical experts or biologists and computer scientists, and that is that there are many cases where the data and the particular biological problem is so intricate and requires so much detailed biological and medical knowledge that it is very, very hard for a person, for a coder, for a statistician to really grasp of what are the important details, of what are the important patterns in a particular complex data set. And all these powerful algorithms that we have and that we are developing, I still find that by themselves they are still relatively blunt instruments, and the ultimate vision is to actually combine the deep human expertise and the deep, unique human, say, cognitive capabilities that we have with the power of big data and with the power of big data analytics. And our best guess for that is what we're trying to build here. So what you see here is, again, Julia Pasmandi, she's a member of my team, and this is a picture that we made using a green screen. So she's wearing a headset and she is standing in front of a massive molecular or genome-wide scale molecular network where every node represents a protein, and these are all the physical interactions, some 500,000 that are known. So this is, you can imagine if you put on this headset that you are standing in a cathedral-sized room, and you can really see all these details at far, or you can, in a very immersive and very interactive way, work with this. And before I actually show it to you a little bit, there are 2 aspects to me that are important of what you can do in an immersive environment, what you simply cannot do at any kind of computer screen that you would ever come up with. And the first one is that you can appreciate network, or data, in our case network structure, across all scales. So you can look at every individual node, you can look at the size of node clusters, let's say dozens or hundreds of them, but you can also really zoom out and in a very seamless fashion explore these massive structures as a whole. And the second aspect of it is that we can also explore them across different contexts. So I will show you in a second that we can inspect what are actually the different kinds of network neighborhoods that are involved in certain biological functions versus what are different kinds of network neighborhoods that are involved in particular diseases, which are generally actually not the same. I think I will actually try it out. So what you're seeing here, I hope you're seeing it, is, well, me putting on this headset and we placed a virtual camera behind me so that you see a 2D projection of what I actually see in 3D. And let's have a look first at what the user interface looks like. It's a very, or what I always thought is a very not so inspiring research field, that is, how do you actually design an interface in an immersive environment? It's actually quite interesting because you want on one hand that you have your, if you want to have a really immersive and very natural 3D environment, you also want the gesture sort of work. So you want that if you want to zoom in, you want something like this, or if you want to push it away, you want to use something like this, or if you want to turn it, you want to use something like this, so that it is actually very, well, what you would naturally do translates also into a functionality of that platform. So if I'm interested in seeing what that node does, I actually want to click on it and then get more detailed information. That said, of course, we are used to these two-dimensional metaphors because we use a computer all day. So we are used that we have a two-dimensional display for data like this, where we can scroll and where we can inspect individual elements. And we try to implement this by using such a, well, stylus type of interface where once I turn my head in a certain way, my controller in a certain way, I can then pretend, or I can then interact with it with my virtual stylus. Okay, so let's zoom into this network a little bit. And I'm not sure, or I guess I am sure, that what you're seeing is quite different from what I'm seeing. Is that in this two-dimensional projection, it still looks rather messy, but if you are in the middle of it, it is actually crystal clear. So I can look into all these different directions of my network and say, okay, there are all these intracellular matrix organization genes that are in one corner. Here we have different kinds of nucleic acid templated transcription. So we have annotated this large network and we can actually do this live. And we can switch between different contexts, as I said earlier, by doing a live relayout, where I can then observe how does the position of a particular gene or group of genes change from a functional context that would emphasize what different normal homeostatic processes these genes are involved in, to a more disease-associated context that actually emphasizes in which diseases is a particular gene or group of genes involved. Okay. Yeah, that is the first thing I wanted to show you. So to get back, two things that I would still like to do in this very short time is to first give you a sense of what could be an application. So what is the first thing that we try to do that is supposed to be clinically meaningful and useful? And the second thing, to give you also some more details on how we actually implemented this. So the first use case that we came up with is to identify and to contextualize rare disease variants. And just a little bit of context, rare diseases are, well, rare by definition. But since there are so many of these rare diseases, they are actually collectively pretty common. So that worldwide, estimations are that roughly every 4 seconds, a child with an inborn, with a hereditary severe gene defect would be born. Now, what is interesting about these diseases from a basic science perspective is that 80% of them are of genetic origin. That is, there is one particular genetic lesion that you can identify that is really causing this disease, but given the amount of genetic variation that we naturally have, the actual challenge is how do we find this one red spot in our genome that is really causing the disease in a particular patient. And next to that, how do we contextualize it? So how do we learn what is the fact that I have an A here where there should be a C? Why does that actually lead to a particular disease that I have? And our collaboration partners that we are working very closely with, they, in order to do this, would go at great lengths for an individual patient to collect all kinds of data that they can pay and that is available and that is relevant to that particular patient, so that ultimately, to us, this is also a paradigm for an omics and data-driven personalized medicine, where I have a particular person and I need, and I'm collecting as much data as I possibly can, and I try to combine it in a way so that I can identify what is the genetic lesion and what is also the phenotypic consequence. To put this again in our network-based framework, the challenge is I have some 10,000 unrelated variants, and I'm searching for that one variant in the genome that is really causing the disease. And in a network-based picture, that would be to find that one red node in the network where all the others are just harmless, unrelated variations. And the strategy to do that is that we first look for the particular network neighborhood that is known to be involved in similar phenotypes, and then prioritize the variants that are close to this particular disease neighborhood higher than those that are farther away. And these is the one specific use case for which we made a particular user interface and for which we work with these clinicians together to really come up with a workflow that would start from preparing the data to getting a first sense of what are the individual variants doing, what is their functional context, to identifying this general disease neighborhood that I mentioned, and then do a detailed inspection of the individual candidates. And by detailed inspection, what we mean is to ask what are the pathways that are involved in them, what are known interaction partners, are any of these involved in similar diseases, are they expressed in relevant tissues, what does the literature have to say about individual genes and their context. So we try to make as much of the functionality that a geneticist expert would really use in order to do this process available. And as time is running out, I would just like to talk also one short slide about how we actually designed it. And there are a few key things. The first one is that we try to design our platform as modular as possible. So we separated the VR-based visualizations from the user interface within the VR. We separated that again from the analytics layer, and that again from the database layer. And the reasons for this are that we would like to enable the community as much as possible to implement their own data, to actually port their own analysis. And since we are using for each of these layers only standard tools, so we use a MySQL database, we use standard Python code to implement the analytics, we do all the user interfaces with pretty straightforward HTML, JavaScript tools to also enable the community again to use what they have been using before and really start developing for the kind of platform without having to hire a game designer. We are in the process of finishing up and cleaning up and polishing our code, so it should be open source soonish, I hope. It is possible to run what I've been showing you is a relatively standard desktop machine with a good graphics hardware, but if you wanted to, by the design, you can also run it in the cloud if you really want to run very heavy computations depending on what your actual application is. And I wanted to give you a link of where you can look at it, but since we are not quite done yet, the only thing I can offer at this point is to follow us on Twitter, and then you'll know if you're interested when you can actually also try and use it and play around with it yourself. So the conclusion is that it is really our ambition to try to instead establish that not as a particular tool for a particular use case, in our case variant prioritization, but to really think a little bit larger and to try to imagine what would be a paradigm for a data analysis, for a data exploration platform of the future. And with this, I think my team, these are the people that actually do the work, and also our collaboration partner, and then of course the people who pay for all this. And again, you for inviting me. I would have loved to be in San Diego right now, but I'm also happy to take questions in Vienna via Zoom. Thanks a lot.
So thank you very much for the very interesting and amazing demo. Do we have any questions from the audience?
Yeah, this is Jake. Jörg, that's very impressive work. So I have two questions. One is, does your software to be released come with some standard map of proteins, either in a interaction coordinate format or some kind of embedding that you optimize,
right,
so the user doesn't have to start from scratch? Yeah.
So when we started that project years ago, we would use standard 3D layouts or Spring algorithms or other things. And what we came to realize pretty quickly is that we need layouts that actually carry an explicit meaning, so that the user knows what does it actually mean that there is a group of proteins that are close by over here, or another one that is very central, or there are some that are peripheral. So what we ended up developing sort of en passant are also specific layout algorithms for this three-dimensional environment. They are pretty straightforward. What we essentially do is dimensionality reduction on a particular feature that I'm interested in. So I could do a dimensionality reduction on, let's say, purely structural network features, such as the centralities or community structures. But I can also do dimensionality reduction on functional features. So I can group genes together that have, let's say, a similar gene ontology annotation, or those that have a similar disease annotation. And we find that it is useful that you actually know what the layout means in order to interpret it. And then you can switch between different layouts to also understand how are different contexts different for the same set of genes.
Okay. So my second question is more philosophical. I like visualization, don't get me wrong. I think that they're extremely useful. We've seen multiple versions of that. But the most useful visualization I see in papers usually zoom in to sufficient biological detail and in comparison between conditions. So I was wondering, with this massively impressive tool, would the metaphor of Google Earth the right killer app, or do you think that you really have to go to the hierarchical organization of the living system, from organs to tissues to cells, and then to proteins in their natural organelle localization to come up with a true killer app, as opposed to the abstract knowledge representation that could be maybe, as I said, trivially represented in detail on 2D paper?
So we still are asking ourselves the same question. And the honest answer is that we actually started from the abstract problem, and that was our immediate candidate to put into our visualization engine. But I myself am not quite convinced that that is the best thing we can do. That said, I think that visualizations that are actually representing something that is not immediately intuitively 3D are even more promising or even more interesting. So to think, how can you interact with an abstract data set, but in a way that is uniquely say, shaped to match the human three-dimensional immersive cognition. So to sort of view the problem from the other perspective and ask, how can I embed this particular data set so that I can work with it better? And I think there is something, say, trying out virtual reality technologies, there is something magic to immersive cognition, really. And the one example that we like to use there is this, it's called the plank game, where you're going up an elevator and you can jump from a skyscraper. And you cannot bring yourself doing it, even though it's, you are wearing headsets and you are in your own office. But there is such a strong link between the visual input that you receive and your cognitive response that I think there is a potential that must be tapped somehow.
All right, thank you.
Any quick questions?
I think there's one in the chat box.
Oh, really? Okay, so Serenan was asking in terms of functionality in discovering biological insight, what is the advantage over cytoscape or navigators? Can it also lay out according to e.g. cell membranes or matrix or nuclear?
So our platform doesn't care what coordinates it receives, so you can lay out whatever you can generate coordinates for. And the most obvious difference between, let's say, cytoscape is that you can work with much, much bigger data sets. So you can actually work with a genome scale molecular network that has 20,000 nodes and 500,000 links. You can look at it, you can turn it, you can zoom in and out. And that is, downright impossible in 2D formats is the most obvious.
Okay, no further questions. Okay, I think in this case, we thank you very much for accompanying with us very late until now.
Yeah,
and it's a very exciting talk, and thank you very much. And then I will try to, let us move to our paper presentation. So our first paper will be scalable Bayesian functional connectivity inference on multi-electrode array recording. And the paper will be presented by Jenny Xu, and Yun Zhao will answer the questions later. So let me share my screen. So please let me know if you cannot hear the audio.
Hello everyone, my name is Jenny Xu. I'm a student from Computer Science Department in UC Santa Barbara. Today, I'm here to present our BIOKDD workshop paper titled Scalable Bayesian Functional Connectivity Inference for Multi-Electrode Array Recordings. In this talk, I will briefly talk about following parts. First, I will talk about biological background. Second, I will introduce our Bayesian framework. Then, the results on synthetic data and a real multi-electrode array recording data. Multi-electrode arrays with advanced neural props have been widely utilized to measure neuronal activities by recording local field potential. I will use MEA in short for multi-electrode array in this talk. Since the same units are measured on multiple recording sites, MEA recordings provide rich spatial and temporal information. The upper left figure is a real MEA device in our lab. The upper right figure is an example of voltage signals. Typically, we do spike detection. For example, we use 6 times standard deviation threshold to filter each signal. Whenever the signal exceeds the threshold, we get a 1, which represents a firing event or spike. Otherwise, we get a 0, which describes as quiet state or no spike. A spike raster plot displays the spiking activity of a group of neurons over time and is widely used to visualize neuronal signals. For this plot, the y-axis corresponds to the index of electrodes, and the x-axis corresponds to the current time in the recording. Based on the spike train matrix, one interesting question is how can we give a quantitative measure of functional connectivity? Researchers tend to build a neuronal connection as a graph. Each node in the graph represents a neuron or an electrode. Edges in the graph stands for the connections. To build a connectivity matrix for the graph, there are some widely used metrics. For example, cross-correlogram, mutual information, maximal information coefficient, and biophysically inspired causal measure. We use a model to first generate some synthetic data, based on which we calculate the functional connectivity using previously mentioned metrics. By using simulated data, we know the ground truth functional connectivity matrix. However, as the figures show, the result gathered from those models looks so different from that of the ground truth. By calculating this cosine similarity, we can verify that those methods are not well known. Maximal information coefficient and Pearson correlation are symmetric, which doesn't represent the real case. Although bio-inspired can get asymmetric result, the cosine similarity is still very low. A data-generated model is required to understand the underlying structure and to make full use of the domain expert knowledge. Bayesian inference can address the requirements for the inference, since it provides distributions for parameters using probabilistic models and observation data. Researchers has proposed a Bayesian model for multi-neuron recordings. However, it is challenging for Bayesian methods to be scalable on large-scale arrays. Given the previously mentioned issue, we propose a scalable functional connectivity inference framework shown in the figure for MEA recording data. And we speed up the expensive Bayesian sampling process through the use of parallel computing. For our probabilistic model, there are three parts: a network model specifying the underlying connectivity of the electrodes, an activation propensity model detailing how a network along with the past spike history affects the probability of the spike at a time being, and the Bernoulli spiking observation model mapping the activation propensity to the observed binary spike trains. The figure shows the probabilistic graphical model for MEA. The model describes how the underlying connectivity of a network of electrodes can lead to the observed spike trains. A considerable challenge for Bayesian techniques is the rapid growth of computation time in accordance with the increasing scale of the network. In the figure, as the number of electrodes increase, the average time for one Bayesian sample increases drastically with the size of the array. The reason is the dimension of parameters to be estimated grow quadratically according to the number of electrodes. To deal with the time complexity challenge, we propose a hierarchical inference procedure. As shown in the figure, we split the whole large array into fixed number of regions. In the first level of algorithm, we perform Bayesian inference individually on each of the smaller regions. In the second level, we treat each whole region as one group by taking the mean of all the spike trains in the region. We apply the same probabilistic model to infer the regional connectivity, which is the connection strength between each pair of regions. The splitting strategy is inspired by the biological phenomenon of regional connections in brains. The splitting strategy decreases the average sampling time quadratically in accordance with the number of subregions. We first use some synthetic data to check the effectiveness of our framework. As shown in the figures, the inferred posterior mean is almost the same as the ground truth connectivity matrix. The inferred functional network adjacency and the weight matrix both achieved a high cosine similarity of 0.99 compared with ground truth. Similarly, we obtain cosine similarity of 0.95 and 0.99 for adjacency and weight matrix when the number of electrodes increases to 10. We also use synthetic datasets to verify the effectiveness of 4 splitting strategies. Here, we assume all the electrodes lie in a line. For the non-overlapping split, we use n electrodes and split them into 2 subregions, front and back, with equal size, and apply the Bayesian inference on each region for the high cosine similarities in both tables. Confirm the effectiveness of our splitting strategy. Here are the results of the regional connections in non-overlapping split. Front indicates the region composed of electrodes 0 and 1 in the ground truth figure, while back indicates the region of electrode 2 and 3. Each element in the inferred regional connectivity matrix reflects the elements of the corresponding regions in ground truth altogether. Regional inferences of the split precisely reveal the overall connectivity between regions. To further verify the effectiveness of our framework, we apply it on 2 sets of real MEA recordings. We make a comparison between our Bayesian inference and ground truth detected by our neuroscience experts. In this figure, green represents the connections found by Bayesian inference, while red denotes the connections not found. 93.33% of the connections detected by the neuroscience experts are detected by our Bayesian inference. We can also see that the real recording has some regional patterns, and the patterns satisfies the intuition of our splitting strategy. We also apply our Bayesian framework to analyze how cadmium affects the structure of neural networks. Cadmium is a toxic heavy metal that accumulates in living systems. Cadmium can change the intracellular concentration of calcium. To analyze how cadmium affects the structure of neuronal networks, we compared the obtained posterior matrix adjacency and weight for a set of MEA recordings, comprised of 1 controlled with no cadmium introduced and 2 other with 1 micromolar and 5 micromolar of cadmium. Baseline recordings were performed 5 minutes prior to any cadmium addition. As shown in the figures, the number of connections and clustering coefficient decreased with a statistical significance with the introduction of cadmium into the culture. The average path length increases significantly with the increase of cadmium concentration. We compared the in degree and odd degree of each electrode for control and 5 micromolar cadmium concentration. We can see that the overall connection pattern maintains, but the in degree decreases prominently with the introduction of cadmium into the culture. Similar phenomenon can be detected for odd degree for each electrode for control and 5 micromolar cadmium concentration. In summary, we propose a scalable functional connectivity inference framework for MEA recording data. We speed up the expensive Bayesian sampling process through the use of parallel computing. We infer the network by splitting the large-scale recordings into smaller local networks. The splitting strategy decreases the average sampling time quadratically in accordance with the number of smaller local networks. We also provide a strategy for inferring the regional connectivity between local networks. This is not only is the computational burden from sampling, but also provides useful insights on regional connections in organoid and brains. Experiments on both synthetic datasets and large-scale real-world MEA recordings show the effectiveness and efficiency of the Bayesian framework. Inference of network structure of cadmium-exposed neuron culture further demonstrates the usefulness of our framework. That's it for this talk. Thank you for your attention.
So thank you very much. Okay. So Yun, are you here?
Yeah, I'm here. Any questions?
So any questions from the audience? No, I have a quick question since you were mentioning about the performance improvement of large-scale arrays. So I'm wondering, what do you mean? How large is the large scale you mean in your presentation, and also how much amount of time you reduce after your improvement?
Yeah, good question. So in our like collaborators lab, we have a collaborator from the biology part. So they have the by large scale array, we mean like more than 2,000 of electrodes. Like, so in that case, we are recording, we have signals from over like thousands of probes. So, for those scales, like most of the actually those Bayesian framework does not scale well. So that's why. But we also find like there are some regional patterns, like the connections does not lies over everywhere. So there are some regional connections there. That's why we propose those splitting algorithm and also we use parallel computing. So because we split it into different regions, so we can calculate like the inner connections separately, basically, and use those parallel computing to boost up the computation.
So eventually, can you reduce the computational time to
quadratically? But I mean,
okay,
right.
Great. So any questions? Any further questions from the audience? If no, then I will just thank Yun because we are a bit behind the schedule, and then I will move to the next presentation. So our next paper, learning prognostic models using disease progression patterns. So Andrea will present the work and also answer the questions. So let me share the videos again. Yeah.
Hello, I'm Andrea Martins with the University of Lisbon, and today I will talk about our work in prognosis prediction in ALS, in particular the use of disease progression patterns to predict the need for non-invasive ventilation. So, let's start with a brief introduction to ALS. ALS is a disease that causes degeneration of motor neurons and subsequent loss of muscle function. While it can present itself and progress in various ways, it is a relatively fast-progressing disease, with most patients experiencing a 3 to 5 year life expectancy after the initial symptoms, and with the most common cause of death being respiratory failure due to the compromised function of the diaphragm. There is currently no cure, with only 2 available medications showing some success in increasing life expectancy. However, non-invasive ventilation, or NIV, has shown the most promising results, as it slows down respiratory function decline, consequently increasing survival time. In this way, it is in our interest to predict when a patient will be eligible for non-invasive ventilation. So, our goal is to ultimately build a predictive model to determine if a patient will require NIV within a specified temporal window, using the test results performed at each patient evaluation. More so, we wish to use the patient's entire evaluation history, rather than, for example, just the last available evaluation. However, because of the heterogeneity in ALS, the amount of evaluations can vary greatly between patients, so for longitudinal data, this would lead to a large volume of missing values. A workaround this issue is to build new temporal features, such as sequential patterns. Therefore, our approach will be to apply pattern mining techniques to our dataset, and use the extracted patterns to assemble new learning sets for different prediction windows. From them, we will build and evaluate several predictive models, as well as look into the patterns themselves. In this work, we use the Portuguese ALS dataset, which contains patient information such as onset age and symptoms, family history and genetic testing, as well as various test results over time. Hence, we have both static and temporal features at our disposal, organized in a 1-row per patient format. Because not all tests are performed at every appointment, we will firstly need to transform the data in a manner that an evaluation is well defined, and comprising of tests performed during a relatively short temporal window. Then, we need to have the data in a way that pattern mining algorithms can interpret. Also, we will only use a subset of features, since some tests have very few available entries and/or are correlated to other tests. So, our approach is comprised of 3 major steps. In the initial step, we build the evaluations themselves and the class necessary for model learning. Next, we construct the learning instances to be used for classification. Firstly, by creating sequence databases and extracting patterns through the SPMF library, and then by building class-labeled binary matrices indicating whether each pattern is verified by a patient or not. Finally, we use these datasets to train and evaluate several classifiers, and also to evaluate the patterns themselves. In the first preprocessing stage, the test results are grouped by date using hierarchical agglomerative clustering, effectively creating an evaluation or snapshot. In these snapshots, each attribute can only be featured once, and all attributes must have corresponding dates where the NIV status is the same. That is, we can't have tests performed before and after NIV was initiated within the same evaluation. The median date of the tests serves as the evaluation date. Then, the NIV statuses of the evaluations are compared to determine the class, which we call evolution. If NIV status changes from 0 to 1 within the specified prediction window, then the patient evolves. If it remains as 0, the patient does not evolve. Snapshots are discarded if NIV was already initiated or there is no future information to determine evolution. As in previous approaches with this dataset, 3 prediction windows were explored: 90, 180, and 365 days. In order to use pattern mining, we need to turn these datasets into sequence databases, and therefore discretize the data. Each feature was turned into several items of the form feature equals value or feature equals interval in the case of continuous variables. The thresholds for discretization were chosen based on previous analysis and according to the clinician's indications. The features were separated into static and temporal for respective pattern extraction, which was done through the SPMF library. The presented algorithms were chosen to extract closed patterns, since they provide the IDs of the transactions or sequences that verify the patterns, which will be necessary at the next stage. 2 algorithms were used for temporal patterns, so that we can assess the effect of temporal constraints. Prefix span only takes into account the transaction order in a pattern, while 4808 also forces the time intervals between transactions to be the same. The support threshold of 24% was selected as it provides a fair balance between the number of extracted patterns and how many patients verify them. Finally, we removed the trivial patterns, that is, the ones with only one item, and build a binary matrix where each row pertains to a patient and each column to a pattern, hence why knowing the IDs is necessary. 1 means the patient verifies the pattern, and 0 means they don't. The evolution value from the last evaluation is also added. This does cause a class imbalance issue, especially for large prediction windows, as most patients will eventually need ventilation. These binary matrices are then used to build and evaluate several classifiers, where we will also use SMODE to tackle the imbalance issue. The matrices are also used to compute the growth rate for each pattern and class. Back to model learning, the classifiers were evaluated through sensitivity, specificity, and AUC score. We can see all algorithms perform quite similarly, with random forests achieving the highest AUC in the majority of cases. The overall tendency is for metrics to improve with increasing prediction window, which can be due to 2 factors. First, the class imbalance becomes more prominent as we increase the prediction window, meaning more minority class tuples are generated through SMODE, and negative examples end up being more similar. Second, as the time window increases, the likelihood for a patient to evolve is higher, meaning there are more positive examples and the probability of similar examples being in the same class increases. When comparing with the best performing approach done with this data, which uses the original features and a specified amount of evaluations, we obtain worse results for the smaller prediction window, but better for the larger ones. As for evaluating the effect of using time constraints, the metrics values don't show any clear distinction, and Wilcox and Seinring's test for the AUC scores has shown there are no significant differences between the 2 approaches, for significance thresholds of 0.1 or less. With this in mind, we would say that sequential patterns extracted through 4808 would be preferable to use, since they perform similarly with a smaller number of features and are more informative and easier to interpret as they specify the evaluation's timestamps. As for pattern evaluation, we looked into the top 20 patterns for each class. As an example, here are presented the top 5 patterns extracted through 4808. In general, the discriminative patterns for the negative class have higher growth rates, which is most likely due to relative support being used. More so, patterns tend to be more discriminative as the prediction window grows. Temporal patterns are in general more discriminative than static ones, and are more present in the negative class, while static patterns are featured mostly for the positive class. Despite this, the pattern featuring ALSFRS between 24 and 36 and phrenic mean amplitude below 0.4, which is temporal and mostly featured for the positive class examples, is the most discriminative pattern for the positive class in all datasets, and the most discriminative overall for the larger time windows. The negative class patterns have a strong presence of items R equals 12 and ALSFRS B equals 12, which reflect unimpaired respiratory and bulbar function, while the positive class patterns include lower scores for these tests. This makes sense, as a patient with strong respiratory function is less likely to require ventilation in the short term. This is also evidenced by the many discriminative patterns for the negative class, which contain R equals 12 throughout several evaluations. While ALSFRS B equals 12 only appears together with the respiratory subscore, lower values show up along with other items, meaning that bulbar function may still be a relevant factor. Phrenic nerve response amplitude may also be relevant, as values below 0.4 only appear in patterns for the positive class, and values above 0.4 in patterns for the negative class. ALSFRS, as well as its subscores for the upper and lower limbs, appear with similar values in patterns for both classes, meaning they may not be as relevant. In summary, we've proposed an approach to predict the need for non-invasive ventilation in ALS patients, making use of their complete evaluation history through pattern mining techniques. The extracted pattern served as features for building and evaluating several predictive models spanning 3 prediction windows. Model evaluation has shown promising results for all prediction windows, and there were few discrepancies between the two approaches used to extract temporal patterns, so using Fournier 08 should be more advantageous. However, we've also seen that the pattern extraction process is highly dependent on the selected parameters and discretization strategy chosen. Therefore, it could be beneficial to explore alternative strategies, including other pattern extraction techniques. Finally, the followed approach does not take into account the heterogeneity found among ALS patients, namely in progression rate. Stratification strategies, as the ones explored by previous work on the Portuguese ALS dataset, can also be implemented, thus generating specific prediction models for groups of patients displaying similar progression. We do need to keep in mind that stratification can be highly impactful to the pattern extraction process, as slow progressors can be expected to exhibit longer patterns and in larger amount, while fast progressors have few evaluations and therefore a decreased likelihood of many patterns being found. To minimize this issue, different prediction windows may also be explored according to progression rate, as they can increase the number of evaluations that can be labeled as well as improve class distribution. Studying these strategies will be our follow-up work. Thank you for your attention.
So thank you very much. Any questions from the audience? So Andrea, are you here? Hello? Hello, Andrea, are you here? Okay, seems that it seems that our presenter did not show up for the question section. Okay, in this case, if you have any questions you would like to clarify, please write them an email. Then let us proceed to the next presentation. So, okay, our next presentation. Okay, let me share the screen again. Hold on. Okay, it's getting large. Okay, so our third presentation. Our third presentation will be a data-driven approach for assessing the impact of reopening order and non-pharmaceutical intervention on the spread of COVID using time-dependent SEIR model.
This talk, which titled A Data-Driven Approach for Assessing the Impact of Reopening Order and Non-Pharmaceutical Interventions on the Spread of COVID-19 using Time-Dependent SEIR Model. This is a joint work with Professor Jeannie Deng, Fauzia Kasai, Hsiang-Yun Li, Nedred Bilo, and Yang Zhao. So I plan my talk to be given as follows. First, the background and objectives, then I will introduce the mathematical models that propose the SEIR model, and then methodology that we used, and I will give numerical results and summary and conclusions. So as we are all aware, the outbreak of COVID-19, which started in early December of 2019, was declared a global pandemic by the World Health Organizations. And since that time, it has been spread around more than 100 countries as of March. And that resulted in both countries' authorities responding to this novel coronavirus by implementing a wide community travel restrictions, lockdowns, as well as facility closures to stop further spread of the disease. due to the little knowledge with regards to the transmission of the disease at the time. And as a result of this action that was carried out by these countries, organizations, as well as authorities, this pandemic caused a global social and economic disruption, including possible largest global recession since the Great Depression. And actually it has affected, causing global famines, affecting over 265 million people. And for this reason, both researchers as well as researchers in both medical and academic fields decided to deep dive into this disease and try to model the possibility to predict what is going to be the possible outcome of this disease. And this is what caused our curiosity to be able to look at this problem. And the objective of this work is to design an SEIR epidemic model for the transmission of dynamics and control of COVID-19 in populations, and secondly, to assess the community-wide impact of early implementation of strict lockdown measures that was carried out in 3 different countries, namely China, Italy, and the United States. And then lastly, to assess the community-wide impact of early lifting of these lockdowns that was in, I think, about 3 jurisdictions in the United States, mainly Texas, Iowa, and Maryland. So we collected the data set from these publicly available sources, which include a World Health Organization, as well as the Johns Hopkins Center for Systems Science and Engineering. And for the data set, for the country data set, China, Italy, and the United States, we collected the data depending on the dates that this pandemic started, or in other words, depending on when the pandemic became a problem to those countries. So we collected for the China between January 15 to May 5, as well as Italy between February to May, and United States between March and May. And then these 3 US jurisdictions, Maryland, Iowa, Texas, because of the lifting of the community lockdown started around April in the United States, and that was why we decided to look at this data from this April 12th to May 4th. So as we know, most countries impacted by the coronavirus pandemic, as of May 16, United States, Spain, Russia, and the United Kingdom share more than 5% of the global cases, which was depicted in this picture. And of course, because of the novelty of this coronavirus and the way countries handle these cases, the pandemic, still the United States was the leading, although that some other countries in Latin America, as well as the Asia, has had significant amount of new cases as of today. And so for this reason, we decided to model, mathematical models in general has been used, has been very influential in providing deeper understanding of the transmission mechanisms and burden on the ongoing COVID-19. Since it's, since around January, both researchers has developed themselves in using mathematical models. And these models has contributed significantly to the development of public health policy and understanding. So these models that has been used mainly of 2 types, which is population-based SIR, as well as agent-based models. So specifically, population-based model has been widely adapted to model the transmission of COVID-19 in a population in that regard. Most recently, a model which was developed by Chen et al. In 2020, which is an SIR model for COVID-19 with the undetectable infected persons, stratifies the population into 3 different categories, mainly susceptible, infectious, as well as recovered. But when we look at this model, it has not really addressed the current dynamic of the COVID-19, which always keep on evolving. And as a result, we decided to propose an SEIR model because of the so our model actually assumed existence of a latent period prior to the onset of infectiousness. So we add one more component to that, one more compartment to that model, which is the exposed individuals in the dynamic model. And then also we included incubation rate, sigma, as well as transmission rate and recovery rate, which we considered them to be time dependent. And then lastly, our model include both symptomatic and asymptomatic infectious persons, which later established in the existing literature that that is the case. And so we obtain, as I mentioned, we stratify our population to subgroups, that is subpopulation, which are the susceptible, exposed, as well as the infected and the recovered individual, and the total population denoted by N of T at time T. And so this is the result. As I mentioned, the sigma of T, which is the incubation rate, the rate at which individuals progress to either symptomatic or asymptomatic patients. That is from susceptible, the rate at which individuals progress from the susceptible state to either exposed, that is from the time they, from exposed to either infectious or with symptoms or without symptoms. So that is what the incubation rate represents. So here is the model, which in this model we stratify the population into 5 different compartments, which is susceptible, exposed, infectious with symptoms, infectious without symptoms, that is asymptomatic, as well as the recovered. So eta represent the relative infectiousness of asymptomatic persons in comparison to symptomatic persons. And alpha is the fraction of cases that are symptomatic. And as such, we obtain the analytical result for this model, for the above model that we just presented. So there is a number which we call basic reproduction number, R0, which for the special case of the above model that I present, when the transmission rate as well as the infection rate, and then gamma I and gamma A are both fixed. And then this, we obtain this reproduction number R0 to be as follows. And then this R0, basically it measures the average number of new COVID-19 infections generated by an average infected individual introduced into the population. And of course, this newly symptomatic infectious persons can infect on average about beta 0 divided by beta I with probability of alpha. And then similarly for the asymptomatic individual can infect on average beta 0 divided by gamma A with probability 1 minus alpha. And so we obtain the analytical result which tells us when this disease goes extinct. So the disease pre-equilibrium, which we denote by this, for the above model is locally asymptotically stable if the basic reproduction number is below unity, that is below 1, and it's unstable if it's greater than 1. So one of the mathematical epidemiological implication of this result is that community transmission of COVID-19 can be effectively suppressed in the community if the control and mitigation strategies that was implemented by the authorities can bring the reproduction number to a number less than unity, that is a number less than 1. And so in order to be able to effectively perform the prediction, we track the transmission rate, the incubation rate, and as well as the recovery rate using ridge regression. So using a method which we call finite impulse response filters in linear systems. And so basically, several machine learning techniques can be used actually to estimate the impulse response parameters that is obtained from these finite impulse response filters, such as moving average, autoregressive models, as well as ridge regression. But we decided to use ridge regression as our estimation method to solve the following optimization problems, which allows us to track this, that is, in other words, to predict this transmission rate, incubation, as well as the recovery rate. And so we implemented an algorithm, which I present here, using a scikit-learn library and grid search cross-validation, which specifically was used to carry out these algorithms, this algorithm using Python programming language. And so that is what we have. So we predicted the number of exposed, infected, as well as the recovered persons. And so here is the prediction results. So the prediction result, as you can see, for the countries, China, between the data which we consider between January 15. So we had, and the prediction window around 3 months, we predicted that the total number of cases will be around 84,413, which is basically similar to what China experienced over the course of the 3 months. And similarly for the United States experienced over the course of this prediction window of 3 months also about 1.7. And that result actually, it's important to point out that this is the case when you have all the mitigation process being implemented were strictly followed by the people and the governments in place. And so also for the 3 US jurisdictions, we have about Maryland, Iowa, and Texas, 33,000, 10,000, and about 47,000 respectively. And this was the case for about 2 months prediction window. And that is the case if entirely the mitigation process that was implemented by those states had been maintained after the lifting of the lockdown restrictions. And so I presented some of the prediction result, which we obtained. And here is the, for the China specifically, the cycle marked solid curved and with dark orange here, red, that is respectively red and green color is the real number of infected persons respectively exposed as well as recovered. And the star marked dashed curve, in this case with light orange, respectively yellow and green color, is the predicted number of infected persons exposed as well as the recovered person. And as you can see, around February, they reach a very steady, sharp increase in the recovered individuals, as well as sharp decrease in the infected exposed as well as infectious individuals. And here is the prediction result for the trajectory of transmission rate, as well as the predicted transmission rate for the China case, and as well as trajectory of the incubation rate and the prediction of the predicted incubation rates for the China case as well. And similarly, trajectory for the recovery rate and the predicted recovery rate for China as well. And here I present the trajectory of the basic reproduction number for the particular case in China. And as you can see, an inflection points which happened around February, because we considered the data set in January 15. And as you can see, after 20 days, they had an inflection point, which is basically tally with what China had experienced around February. They were able to have the reproduction number below 1, and that they were able to contain the transmission of the disease. And so this is, so as you have seen that our model was used to assess the community-wide impact of early lockdown in China, in Italy, and the United States, as well as various levels of lockdown lifting in the United States. That is in the 3 jurisdictions in the United States, mainly Maryland, Iowa, and Texas. Thank you very much. And if you have any question, our team is ready to answer your questions. Thank you very much.
So thank you very much. Do you have any questions for the author? So is Isma here? any co-author of this paper?
Yes.
So do we have any questions from the audience? No, so okay. So maybe I didn't understand completely. So could you briefly explain what is the limitation of your approach?
Hello, what is the word?
What is the limitation of using this approach? Because now you are predicting, for me, the result looks quite promising. So is there any limitation for that?
Yeah, it's just when we are getting the number of exposed people, because all the data set we had, there wasn't exposed people. So we have to use an idea in one of the papers to kind of simulate those people.
Okay. Okay. No further questions? I have a feeling people are very quiet and shy today. Okay. Okay, if there are no further questions, maybe we move to our next presenter. And thank you very much, Fauzia. And okay, and thank you. So our last presenter will present assessing reliable COVID-19 infect numbers, the Italian case. And okay, and I think Pirangelo, you will present your work, right? So yeah. I will assign you the right to share the screen.
Okay.
Okay, you should be able to share the screen now.
Okay. Okay. Can you see the screen now?
Yes.
I guess so. Okay. So first of all, thank you very much for your time. It's the letter of the session, so I will try to gather your attention, and I will avoid some formulas. So this is an abstract paper, so we will focus mainly on our experiences about Italian cases. So we started working on end of January about the situation in Italy. Some of the concepts I'm going to present have been treated by the previous presentation, but of course during other presentations. So let me please introduce a couple of snapshots. So first of all, what about the real numbers of COVID-infected persons? So this is strictly related to the number of swab tests. The more swab tests you perform on population, the more you are reliable in terms of asymptomatic and in terms of real pictures that you have of a nation. Second items, the lockdown, real lockdown, strict lockdown is the only condition and position that is able to maintain the diffusion of COVID-19. And third point is the perception of the exponential growth. So these are three main points that we should focus, especially looking at data. What is the latter one? The latter one is regarding the fact that if you discuss with common people about exponential growth, okay, the citizen and normal people has no perception of what is exactly the exponential one. What is the example? Something like I give you $1 and I say that in a bank account is going to increase exponential. So how many times do you need to have millions of dollars on your bank account. In the exponential case, this is something in terms of days, something like 60 days, something like 3 months. So this is a perception that is completely missing in our pandemic situation. Okay, so let's go further, and I'll show you what are our experiences. This is a joint work with two colleagues of University of Catanzaro in south of Italy, and with Tamer Kaveci, that is a colleague of Florida, University of Florida. So please look at this picture. On the top part of the presentation, we have Pantheon, that is the center of Rome, Via del Corso, that is the center of Rome, and the pictures that are downstairs, that seems to be especially the two ones here, Pantheon and Via del Corso, are pictures on March. So you know what it means, having completely empty Rome. This is the real lockdown. That is something that in Italy has probably reduced the numbers. And let's look at the numbers. We focused on numbers, for instance, in April 2020. In April 2020, we have something like more than 100,000 victims and almost 20 in Italy. What about the numbers? Numbers was about 2 million of infected persons. And of this one, the infected one in Italy were in terms of thousands. What happens in August? Now in August, we have one order in the world, one order. We have 21 million. But in Italy, we have almost less than 300,000. So it means that the lockdown, even if we have an increasing number, the action that we got it was able to maintain data in the same order. We passed from 2 million to 20 million. In Italy, we passed to 100,000 to 200,000. So this is in terms of numbers. Let's put on the question we considered. So we considered mainly two main topics. And both topics have been analyzed with respect to geographical scaling down analysis functions. The first things is focused on what are the real numbers of infected people. And in particular, in Italy, we had the Lombardia region, that is the north part of Italy, has been infected much more than the whole country. So we scaled it down in terms of, you know, divide et impera. So something like you go and deep and you try to see what happens at the regional scale. And so here we have the growing in Milano, Brescia, Bergamo, that are single cities. So first of all, which are the real numbers? And the other things is something regarding the, as we will see, the number of places in intensive care units. But most of all, as I told you at the beginning, we have to focus about terms. We have to focus about strategies. We have to focus in February about what are the different keywords that we should manage. So the first topic is about number of assessment. The first observation was that the number of deaths in Italy was too high with respect to the infected and the number and the population. And the other one is related to what is the necessity? Why in some places you have this too high number of deaths? And this is related to the non-scalable places in terms of intensive care units. In particular, as you will see here, you have a distribution in Italy of the, in blue, you have the number of beds or in intensive care units that have been occupied, and the other one in green as the availability. As you can see, in many places, here is Sicily, of course, you have a high value of availability, but you have low number of ICU requirements. In Lombardia, that is the northern part, you have in March that you have requirements to high number of intensive care units and respiratory masks. So the importance is about gathering what are the real number of infected, and we try to predict in terms of 10 days, no more than 10 days, but this number of 10 days followed the health structure in moving persons from and healthy people from one health structure to another one, even among regions. We focused on the case fatality rate, the CFR, that is the percentage of death node coming from the viruses of the same family. We consider the history of some similar structure viruses, and we consider the case fatality rates evaluated in March. We obtained the case fatality rate in Lombardia region that was almost 20%. That is a too high value with respect to the number of measured infected citizens. So we started some hypotheses, and we considered the 3 or 4 possible scenario in which we have 5–4% of case fatality rates, and we obtained some reliable, much more reliable numbers in terms of asymptomatic and infected person that were a number able to justify the rapidity in which number growing so in these terms. We those was able to consider the increasing numbers in terms of time windows of 5 up to 10 days, but this number this time window is very large if you consider the exponential phase of the infection. Of course, we did this in terms of whole country, but most of all, we did it in terms of regions. So, for instance, we have an observation of in terms of officially measured almost 50,000 infected people. In reality, by using our CFR, we arrived in terms of 5 or almost 300,000 infected. And these numbers are much more reliable with respect to what we measured in terms of infected and in terms of rapidity of the diffusion. And of course, this was observation was confirmed by the observation in the days later on timing, because we worked on a time window of almost 1 month. And most of all, we considered what happens in regional places, starting with the containment measures. Why containment measures? Because the containment measures has been implemented in Italy in terms of single places. For instance, we have this place Codogno, that is a little country that was locked down, and everything was nobody could come in, nobody could come out from this little city. And then we have also a red zone in regions of North Italy, Lombardia, Emilia, and Lazio. And now you will see this is a time scaling diffusion. You see that before the lockdown phases, you have this blue line that has a slope that is with an increasing number. Then you have the red, and after 10 days of lockdown, you have this yellow increasing number of diffusions. And finally, the numbers that we gathered was online with the requirements in terms of recovery. And most of all, the more we went in details in terms of regional scale, the more we were precise and in predicting values. the similar strategy has been used in terms of predicting the number of resources, and we focus that in particular on respiratory, or in terms of bed resources for intensive care units. We started from the number of available bed resources that are divided by the 20 regions that are government administration, in which is divided the Italian country, and we consider the infection number per days and the requirements of this infected person, the percentage of the infected person that required the recovery in intensive care units. We applied such model for single regions. And so what we did is something like that. On the left part, you have the regional diffusion of infection in the regions. And on the right part, you have, we put on the same graph, we put the number of infections versus the number of ICU beds that are occupied. And with a prediction of 5 up to 10 days by using the interpolation lines on the right part of this graph, we were able to predict the number of ICU. Of course, we tested this analysis by, the timing we wrote down our model, we obtained it, then we measured the data, observed the data furnished by the Italian ministry. In conclusions, we are still working on analysis and data models. We also try to put all together the observations that we provided in terms of clinical as well as in terms of molecular models, and we have a joint work with some colleagues from the Johns Hopkins University and with the colleagues who are abroad, that is the colleague, an Indian colleague that is working in USA currently. Up to that I stayed in time, so thank you very much for your attention.
So thank you very much for the interesting talk. Do we have any questions from the audience?
I was just curious, where did you get the information for the ICU beds?
Well, the Italian structure, the Italian health structure is managed in a central way.